Hello all, I am Dr. Gauri Abhyankar, working as Assistant Professor in Abasaheb Garware College, Pune. This session consists of study of nucleus, the second part of it. Here we are going to cover the nuclear import and export of various molecules through nuclear envelope. Now, as you all know, nuclear envelope is a punctate structure which consists of large number of nuclear pores. These nuclear pores would carry out the import and export of various biomolecules through mainly two processes. One is simple diffusion, which is a passive transport, and the other is selective active transport. Now, the smaller molecules can pass through nuclear pore through simple diffusion. Those molecules which are usually less than 60 kilodaltons in size can go for passive transport, whereas larger molecules would require selective active transport. Now, this selective active transport is very important and crucial in all eukaryotic cells. The selective transport would carry out or would require large number of receptors. Now, large number of proteins would be transporting either from nucleus to the cytoplasm, or they are also transported from cytoplasm to the nucleus. So, this session would be done with the learning objective as nuclear import and export of various molecules across the nuclear envelope. All of us know that many different proteins like histones, DNA and RNA polymerases, various transcription factors, splicing factors are imported into the nucleus from cytoplasm. The transport is a two-step process. In first step, soluble import receptors in the cytoplasm recognize specific proteins to be imported in the nucleus and form a protein receptor complex in the cytoplasm. The next step is subsequent transport of the protein receptor complex into the nucleus through nuclear pore complexes. The proteins to be imported contain a specific amino acid sequence called as nuclear localization signal. These signals are recognized by the transport receptors, and only those proteins which have these signals can pass through nuclear pore complex. The slide shows you two different types of nuclear localization signals. The first nuclear localization signal was characterized in simian virus 40, that is SV40. The nuclear localization signal in SV40 was 7 amino acid sequence necessary for nuclear transport of T antigens. The other nuclear localization signal that you are observing in the slide is of nucleoplasmin. It consists of two parts. One part is a pair of lysine arginine and second part is 4 lysines. Both these parts are 10 amino acids away from each other, but both are necessary for nuclear transport. The slide shows you the overview of import of various molecules through nuclear pore. Most of the nuclear transport proteins are members of karyoferrin protein family. The transport across nuclear membrane is facilitated by RAN cycle. RAN is a small GTP binding protein that shuttles between cytoplasm and nucleus. In cytoplasm, importin alpha, also known as karyoferrin alpha, and karyoferrin beta bind to the nuclear localization signal on the cargo molecule and forms a cargo importin complex. Cargo molecule is the molecule to be imported. The figure shows you importin alpha as green circle, karyoferrin beta as orange circle, and cargo molecule containing nuclear localization signal is shown in blue color. Once the cargo importin complex is formed, it passes through the meshwork of nucleoporins. When it reaches the nucleus, then importin beta from this complex binds tightly to the RAN GTP present in the nucleus. This binding causes change in the conformation of importin. Due to this conformational change, cargo molecule dissociates from cargo importin complex. The importin beta RAN GTP complex then leaves the nucleus so that importins return to the cytoplasm. In the nucleus, high RAN GTP concentration is maintained by RAN GEF, which is RAN guanine nucleoside exchange factor. It is an enzyme that stimulates conversion of GDP to GTP. Whenever importin RAN GTP complex reaches the cytoplasm, RAN GTP undergoes hydrolysis and becomes RAN GDP. Once RAN GDP is formed, importin can no longer bind to it and so it is released. The importin is now free to go again and form complex with another cargo molecule. RAN GDP has its own import receptor known as NTF2. This NTF2 transports RAN GDP to nucleus so that RAN GTP is regenerated in the nucleus. Here you can see the export pathway showing transport of various molecules from the nucleus to cytoplasm. Cargoes or proteins that are destined to be transported from the nucleus show a nuclear export signal. You can see cargo proteins in blue color again. Such proteins form a complex with CRM1 and RAN GTP before they are exported out of the nucleus. CRM1 is shown as green circle and RAN GTP as violet rectangle in the figure. These complexes pass from the nuclear pore complex and reach the cytoplasm. In the cytoplasm, RAN GTP is hydrolyzed to RAN GDP by RAN GAP that can be seen as yellow rectangle in the figure. RAN GAP is RAN GTPase activating protein. It is an enzyme which stimulates hydrolysis of GTP to GDP outside the nucleus. Therefore, the cytoplasm shows high concentration of RAN GDP. Hydrolysis of RAN GTP to RAN GDP helps dissociation of the cargo protein CRM1 RAN GTP complex. CRM1 is then transported back into the nucleus where it can reassociate with a protein containing nuclear export signal and RAN GTP to start the process once again. Note that RAN GAP is localized in the cytoplasm. So, cytoplasm shows high amount of RAN GDP. On the other hand, RAN GEF is present inside the nucleus. So, nucleus shows high amount of RAN GTP resulting in unequal distribution of RAN GTP across the nuclear membrane. The concentration of RAN GTP determines the directionality of the nuclear transport of cargo molecules. RAN regulates movement of cargo molecules by controlling the activity of nuclear transport receptors. In plants, RAN GAP is associated with nuclear envelope during interphase. It contains plant-specific N-terminal domain called as WPP domain. This domain is unique to plants and is not observed in animals or in yeasts. It shows strong similarity to the unique plant protein called as MAF1, which is a small nuclear envelope associated protein. Arabidopsis expresses 5 WPP domain proteins. These are RAN GAP1, RAN-GAP2 and 3 short proteins, namely WPP1, WPP2 and WPP3. The WPP domain is a unique domain necessary and sufficient for targeting to the plant nuclear envelope. In figure A, Arabidopsis RAN-GAP appears at the emerging cell plate in dividing plant cells during anaphase. It can be observed as green fluorescence at the cell walls in figure B. It remains associated with the growing rim of the cell. The figures also show red stained nucleic acids. WPP domain interacting protein 1, that is WIP1, binds RAN-GAP as well as WPP domain. These WIPs target RAN-GAP to the nuclear envelope. The white arrow in last photograph shows you targeting of RAN-GAP to nuclear envelope so that the envelope shows more fluorescence due to concentration of RAN-GAP. In addition to transport through nuclear pores, RAN cycle is crucially involved in assembly of nuclear pore complex, in regulating spindle formation, kinetochore attachment and nuclear envelope assembly during mitosis. The slide shows cyclic model for transport of RNA polymerase II. RNA polymerase II transcribes protein coding genes in the nucleus of eukaryotic cells. The enzyme has 2 large subunits and 10 to 15 smaller subunits. Because these polymerase II subunits do not have known nuclear localization signal, the exact mechanism of its import into the nucleus was not very clear till recently. Now, studies have come up with a cyclic model for nuclear import of this enzyme. First, polymerase II assembles from its subunits in the cytoplasm. As you can see, there is a cleft in the assembled polymerase II. Then, a protein IWR1 binds in the polymerase cleft and completes the assembly of polymerase II. IWR1 is a 40 kilodalton protein having a bipartite nuclear localization signal, that is, one nuclear localization signal for import and one nuclear export signal. The IWR1 nuclear localization signal directs import of polymerase II through nuclear pore complex with the help of cap 60 and 95. Once inside the nucleus, polymerase II associates with transcription initiation factors. This association releases IWR1. Nuclear export signal on IWR1 directs its export to the cytoplasm and completes the cycle. But note that IWR1 function is polymerase II specific and transcription independent. Transcription factors are functional only when they are present in the nucleus. So, regulation of their import is very unique way of controlling gene expression. The figure shows you 2 mechanisms of regulation for import of transcription factors. In one mechanism, transcription factors or other proteins like NF-kappa B associate with cytoplasmic proteins that mask their nuclear localization signal. In the figure, you can see I-kappa B as an inhibitory protein masking the nuclear localization signal of NF-kappa B and forming an inactive complex. So, its transport to the nucleus is prevented. If this I-kappa B is phosphorylated and degraded by ubiquitin mediated proteolysis, then the nuclear localization signal is not masked and NF kappa B can be transported to the nucleus. In other mechanism, as in yeast transcription factor FO4, the transcription factor gets phosphorylated at a typical serine residue adjacent to its nuclear localization signal. This phosphorylation renders FO4 inactive and inhibits nuclear import of FO4. Whenever regulated dephosphorylation of this site takes place, it activates FO4 and then it can be transported to the nucleus. Next few slides will describe nucleocytoplasmic trafficking of proteins. This protein trafficking is a highly selective process and is very important in plant signaling pathways. There are many pathways involving nucleocytoplasmic trafficking, but none of them are mutually exclusive. These pathways can be grouped into 4 broad categories. This slide shows the first category, which consists of protein modification or conformational change that either masks or exposes nuclear localization signal or nuclear export signal. The figure on left shows regulation of phytochrome B and phytochrome A. Phytochrome B is a light stable dimer. In dark, the nuclear localization signal is masked, which you can see as 2 blue petal like structures. When phi B is exposed to light, absorption of red light brings about conformational changes in phi B, opening up the dimer and exposing the nuclear localization signal. See the change in the direction of blue petals. Once the NLS is exposed, phi B is imported, whereas phi A import requires interaction with plant specific cytosolic factor FHY1, which contains nuclear localization signal. Whenever the plant receives red light, FHY1 gets associated with phi A and allow its nuclear import. The figure on right depicts redox mediated regulation of defense gene expression NPR1. Many times innate immune responses are associated with changes in cellular oxidative states. NPR1 localization responds to these redox changes. In non-stressed plants, NPR1 accumulates in cytosol and undergoes oligomerization or large complex formation. After pathogen attack, salicylic acid concentration increases. As a result, NPR1 monomers are released from the complex and are transported to the nucleus. Here you can see the second category of pathways where nuclear import is carried out by heterodimerization or heterocomplex formation. It plays major role in plant pathogen interactions. The left hand panel shows you proposed nuclear import by diversion from chloroplast targeting. In uninfected plants, the chloroplast protein NRIP1 is located in chloroplast and the other resistant protein N is located in cytoplasm. So, the resistant protein does not enter the nucleus. Whenever there is viral infection like that of tobacco mosaic virus, viral effector protein P50 is present in the cytoplasm. Now, NRIP1 forms a complex with P50 and the resistance protein N in the cytoplasm, and this complex then enters the nucleus. The right hand panel shows you regulation by heterocomplex formation in Arabidopsis, circadian clock factor TOC1 and PRR5. Both show peak expression during early evening and are able to heterodimerize in vivo. A large proportion of PRR5 is always present in nucleus independent of TOC1 levels. But when PRR5 is overexpressed, TOC1 is concentrated in nucleus. So, PRR5 facilitates or helps both nuclear accumulation and hyperphosphorylation of TOC1. The slide mainly focuses on last category of pathways, that is cytoplasmic retention by membrane association and its release by proteolysis. Again, see the figure in left hand panel. Heat stress induces proteolytic release of BZIP28 from membrane of endoplasmic reticulum and its redistribution to nucleus. Figure in the right hand panel shows regulation by 14-3-3 interaction. GA3 encodes Nt-kaurin oxidase that acts early in gibberellic acid biosynthetic pathway. A transcription factor RSG activates gibberellic acid. In absence of gibberellic acid, RSG distributes equally between cytoplasm and nucleus. When gibberellic acid is applied, RSG is rapidly partitioned in cytoplasm through phosphorylation and subsequent binding to a 14-3-3 protein. This response results in gibberellic acid mediated down regulation of its own biosynthesis. Similar mechanism is observed in brassinosteroid signaling. Phosphorylation of transcription factor BZR1 promotes binding to 14-3-3 protein, causing cytoplasmic retention and or nuclear transport. Pre-60S ribosomal subunit undergoes a complicated series of assembly steps in the nucleolus and reaches cytoplasm. It recruits several different classes of export receptors. The CRM1 export receptor binds in the presence of RAN-GTP to the nuclear export signal containing adapter NMD3. The other export receptors MEX67 and MTR2, also ARX1, can bind directly to the pre-60S subunit. The large complex is then translocated through the nuclear pore complex. After its transport, the export factors are removed from the pre-60S particle in the cytoplasm. Once RAN-GTP hydrolyzes to RAN-GDP, CRM1 dissociates from NMD3. NMD3 is released by LSG1 GTPase and is replaced by the ribosomal protein RP110. ARX1 might be dissociated by the release factor REI1. Export receptors are seen as yellow, adapters are seen as blue, and dissociation factors are shown in violet color. Eukaryotic cells export many different classes of RNA molecules from the nucleus to the cytoplasm, where majority of them participate in different aspects of protein synthesis. For all classes of RNA molecules, nuclear export depends on the assembly of RNA into appropriate ribonucleoprotein complexes. The slide shows major routes for RNA export, that is export of tRNA, microRNA, small nuclear RNA, mRNA and ribosomal RNA. Processing for each shows primary RNA transcript as well as the transport competent RNA after it has undergone processing, maturation and assembly with export factors. Export adapters are shown in blue, whereas export receptors are shown in yellow. Prominent structural motifs in pre-RNAs are indicated. For the mRNA export route, the names of both metazoan and yeast proteins are indicated, and mRNAs are shown with additional adapter proteins and RNA binding factors. The figure shows nuclear export of mRNA, which is a very complex process. The nascent transcript generated by RNA polymerase II is co-transcriptionally folded and associated with TREX complex to assemble into a pre-mRNP, that is pre-messenger ribonucleoprotein. The mRNP associates with additional pre-mRNA factors and RNA binding proteins. After this, mRNA export receptor is recruited to the mRNP via adapter proteins such as YRA1. When emerging at the cytoplasmic side of the nuclear pore complex, mRNP encounters a remodeling machinery on the nuclear pore complex fibrils that consists of ATP dependent RNA helicase and its activators and the signaling molecule inositol hexokininophosphate. After dissociation of MEX67 and MTR2 and other export factors from the RNA cargo, mRNA proceeds for translation. So, let us summarize quickly what we have seen. Initially, we started with nuclear export and import through RAN cycle. Once we have seen the RAN cycle for export and import in detail, then we move to the role of RAN GAP in plants and its cellular localization. After that, we have seen the cyclic model for RNA polymerase II transport with the help of IWR1. After the cyclic model for transport, then we have went to see the regulation of nuclear import of transcription factors. We have seen two pathways for regulation. Then we have seen in detail the nucleocytoplasmic trafficking of proteins and 4 different categories of pathways which are involved in this nucleocytoplasmic trafficking. And last, we have also seen the nuclear export of different RNA molecules from the nucleus.